Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So thank you so much for joining my session this evening. So today we are in lecture 3 of photosynthesis, so photosynthesis and higher plants. So we have been talking about PS1, PS2. So what is cyclic photophosphorylation? Sorry, we didn't talk about cyclic photophosphorylation. If I remember well, we stopped with the Z scheme. So we are supposed to talk about chemiosmotic hypothesis and non-cyclic photophosphorylation today. So that is our topic for today. So I hope you are enjoying. I hope you guys had a very great weekend, right? So can we go? Yes. So I'll just finish off with two concepts today. So I'll teach you, I will revise what is that scheme, what is 𝐿-HC complex. So today we'll also talk about what is a quantasome, and then we will talk about cyclic, non-cyclic photophosphorylation, what is the difference between the two, and then we will see what is chemiosmotic hypothesis. So this is a hypothesis where a lot of students get a lot of doubt him. Ma'am, we seem to understand, but we don't understand actually. So that is the tricky part in this chapter, I would say. It's a hypothesis because scientists believed it, that is the way it is happening. So we don't know if that is the way it is happening, okay? Yes, let's move in and let's have a quick recap of what I spoke in the previous lecture. So that will give us a continuation because we last met on Friday, if I remember. Yes, let's continue. Here we go. So we are done with a lot of experiments, I guess. So so many experiments we have read. So yeah, so we are right here exactly. The cyclic and non-cyclic photophosphorylation. Then we are going to talk about chemiosmotic hypothesis today. All right. So giving you a recap, so I told you when light falls, so there's something, so we were talking about the pigments, right? So if you remember, there are 4 pigments that is involved in photosynthesis. So can you quickly tell me what are the 4 pigments that is involved in photosynthesis? Quick? Yes. So the first pigment, so I'm talking about the pigment that is involved in photosynthesis. So the first pigment is chlorophyll A. So the second pigment is chlorophyll B. Then you have xanthophylls and carotenoids. Right? So this is our chief pigment. These are our accessory pigments. Fine? Okay. So this chief pigment, because it can trap maximum light, right? And the accessory pigments help in trapping the light of the chief pigment, and it also prevents the chief pigment from getting photo-oxidized. So just give me a minute. For all those who are watching my lecture, so thank you so much. So please don't forget to like, share and subscribe. Right. Yes, ma'am, a very happy good evening. Hi, W, a very happy good evening. So sorry, there's a technical glitch. I couldn't look into the monitor. So I just turned on my phone to look into the chat box. Yes, a very happy good evening. So thank you so much for joining my lecture this evening, and please don't forget to like, share and subscribe. And sorry for responding late, okay? Yeah. So we know the chief pigment, okay, so we know light is trapped by light harvesting complex, that is 𝐿-HC. So inside the light harvesting complex, we have the reaction center. The reaction center has your chief pigment, that is chlorophyll A. Right? So here we have the antennae, which has the accessory pigments. Right? So this 𝐿-HC is organized into systems that is called as the photosystem 1 and the photosystem 2. So these photosystems are named in the order in which they were discovered, not in the order that they work. Right? So how is that scheme? So let's have a quick drawing here so that you understand. So light falls here. So light falls on the 𝐿-HC. So first actually your PS2 is activated, so which has a wavelength of P680 nanometer. Fine. So once light falls, so all the electrons are excited here and the electrons move to the excited state. So here we have the primary electron acceptor. So the primary electron acceptor here is your ubiquinone. Fine. So from here the electron further moves into the electron transport system. So when it moves inside the electron transport system, you have the release of ATP from ADP plus Pi. Now the electrons move into plastocyanin, which is a copper containing complex. So from here it moves into the LHC of your PS1. Actually light also falls on the LHC of the PS1. So PS1 is at a wavelength of 700 nanometer. So again the electrons are excited here. So once the electrons are excited, the primary electron acceptor here is your iron sulfur complex. So again the electron moves downhill. When it moves downhill, so you have the reduction of your NADP to NADPH plus H plus. So where is this entire process happening? So this entire process is in the shape of a Z. So it is also called as the Z scheme. So I also told you Z scheme is also known as non-cyclic photophosphorylation. So where is this happening? It is happening inside the thylakoid membrane, because it is a part of your light reaction, if you remember. So ATP is produced as a product of your light reaction. NADPH is produced. Yes? Okay. Yes, xanthophylls and carotenoids. Alright, so I can see your messages now. Very good. Okay, fine. So here, how will the electron stock be replaced once again? So the electron stock will be replaced by the splitting of water molecules. So water molecules splitting associated with PS2. So when the water molecules split, you have the electrons which gets replenished and your H plus remains inside and O2 is a product of your light reaction. So this is how your Z scheme happens, right? So I hope it's clear. So now we move forward to the next part. So how does it further happen, right? So there's something called as cyclic photophosphorylation. So in cyclic photophosphorylation, what happens is that, so you have your light. So light falls directly on your LHC of your PS1, so which has a wavelength of P700 nanometer. So once again the electrons are excited. So the primary electron acceptor is your iron sulfur complex. But in your cyclic photophosphorylation, what happened? You send it sometime before and you send it now. Okay, fine, alright. So now what happens? The electron does not come and help in the reduction of your NAD+, but instead it cycles back to the ETS. So when it cycles back to the ETS, your ATP is produced from ADP plus Pi. Then it goes to plastocyanin and it comes back. So the electron keeps cycling. So this is why it is called a cyclic photophosphorylation. So also please understand, in cyclic photophosphorylation, only PS1 is involved. So PS2 and your NADPH, sorry, it is NADP reductase. Your NADP reductase are absent. So there is no splitting of water here because there is no PS2, because splitting of water is strictly associated with PS2. So there is no PS2 here, so there is no splitting of water, right? And this happens in the stromal lamellae. Fine. So now we have understood the beauty of light reaction. So once again, what quickly happens in the light reaction? So in the light reaction, your light is trapped. Who traps the light? That is why we have your LHC. And inside the LHC, we have two complexes. What are they? So what happened? What happened, W? You're not feeling well today. So why is that you have sent an emoji with a thermometer inside the mouth? So you are having fever, you're having high temperature. Is that so? Okay, Raghuram, hello, hi. So you have sent something and then you have deleted it. Yes, hello Raghuram, a very, very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Okay, so let's continue. Let's continue here. So light is trapped by the LHC. The LHC has two components. I hope you remember, reaction center and your antennae. So once light falls on your LHC, the electron gets replenished. So PS2 is associated with the splitting of water. So once the electron moves forward, it produces your ATP as well as NADPH. Right? Now the light reaction is done. Now we have to talk about the dark reaction. But before we talk about the dark reaction, how is the ATP produced? There comes the next question. Okay. Okay, your mouth has pen, so that is why you send it with an emoji. Okay. So you are going to be a future doctor and you say your mouth has a pen. So is it that not a childish habit? Yes. So don't put your pen in an open space so that it can catch more microbes, and if someone touches it by mistake, so that leads to the spread of communicable diseases. So please don't do it, W. Yes. Alright. Okay. Now the next part is scientists wanted to know how the ATP is produced. Okay. Okay, ATP is produced when the electrons move and the electrons move into the electron transport chain. The ATP is produced. Exactly how? I just write like when the electrons move inside the ETS, I have the production of ATP. How it can be produced? So scientists actually hypothesized a way as to how it can be produced. It's a hypothesis because they are assuming it to be so. They are not very sure, so you cannot break open a cell, we cannot break open a thylakoid and say, hey, do your process now, I want to overlook. That's not possible, right? So scientists hypothesized that this is how it is going to be, and that is why we have the chemiosmotic hypothesis. So now we are going to talk about the chemiosmotic hypothesis. Ashutosh Kumar, hello, hi, a very, very happy good evening, and thank you so much for joining my lecture this evening. So please do like, share and subscribe. Alright, so now let's see what is your chemiosmotic hypothesis. That is the most important topic for today. So cyclic photophosphorylation, when the PS1 alone is functional, okay, and the electrons are circulated within that PS1 photosystem itself. Alright, so phosphorylation occurs due to the cyclic flow of electron. Phosphorylation is addition of phosphate group. So it occurs in the stromal lamellae. Membrane or the lamellae of the grana has both the PS1 and the PS2. That is, within the thylakoid membrane, you have PS1 as well as PS2. PS1 is towards the end of the membrane. Alright, stromal lamellae exactly lacks PS2 plus the NADPH reductase enzyme. So the excited electrons cannot pass downhill for the reduction of NADPH. So the cyclic flow results in the synthesis of ATP. The cyclic photophosphorylation occurs only when the light of wavelengths beyond 680 nanometer is available for excitation. Very simple, because here the wavelength is P700. So it has to be beyond P680, right? So this is how your cyclic photophosphorylation happens. So remember, the electron acceptor here is your FES complex. Fine? Okay. So now coming to the chemiosmotic hypothesis, the most important concept of today's talk. So how actually ATP is synthesized in the chloroplast? So what is the exact mechanism? So chemiosmotic hypothesis explains how ATP is produced. So that is the reason we are studying this mechanism. Alright. So before we go to chemiosmotic hypothesis, I want you to remember what is a quantosome. So I should have told about quantosome much before, probably in the last class, but I am telling it to you now. So please pay attention. Quantosome is the number of pigment molecules that is required to produce a desired photochemical reaction. So that is called as a single photosynthetic unit or a quantosome. So for example, there should be a minimum number of 2 to form a team, right? So the minimum number of pigment molecules that is present to initiate or start a photochemical reaction is called a quantosome or a single photosynthetic unit. Alright. So one unit consists of approximately 230 chlorophyll molecules plus accessory pigments or the harvesting pigments. So the accessory pigments are also given the name harvesting pigments. That is, in a light harvesting complex, for the photochemical reaction to activate, I need a quantosome. Inside a quantosome, I should have at least 230 chlorophyll A molecules plus other accessory or harvesting molecules. So in each group or unit, there is a special molecule that is very, very important. That is your reaction center. The others are present in your antennae or your harvesting pigments, that is the accessory pigments, right? So a typical quantosome is here. Alright, fine. So now coming to the concept of chemiosmotic hypothesis. So they are hypothesizing that this is how the ATP is produced. So you should understand, whenever ATP production is there, it is associated with a proton gradient. So what is a proton? Proton is a H plus gradient, right? So when I say proton, proton means H plus. Gradient means difference. There is one place there is high concentration of protons and another place there is low concentration of protons. That is a gradient. So gradient is a difference. Fine. So that is what happens. So whenever we are talking about the production of ATP, there should be protons and that proton should form a gradient. Then that gradient should break. That is what leads to the synthesis of ATP molecules. Alright. Okay. In photosynthesis, inside the thylakoid, so if this is my dish-shaped thylakoid membrane, the space inside this is lumen. Right? Yes? H plus through? No, it comes here also. ETC may H plus comes in respiration. That is in the next chapter. This is photosynthesis. Okay? In photosynthesis, H plus comes between the lumen and the stroma. So the space inside the thylakoid is lumen and the entire space of the chloroplast is stroma. Right? So the H plus concentration is more here and the H plus concentration is less here. So H plus is there. The gradient is, now there is a gradient, here there is more concentration, here there is less gradient, right? So now this difference in concentration will be broken. When it is broken, it will give you your ATP molecules. We are going to see how it happens. Just like W reminded us in ETC also we have a proton gradient. We will see that when we are talking about respiration, right? So now, when I said the lumen of the thylakoid has more amount, okay, so see here, so you have the stroma, you have the lumen, right? So according to the chemiosmotic hypothesis, so the lumen has more of H plus ions and the stroma has less of H plus ions. So a difference in concentration is set up. But now comes another question. How is that the lumen has more of H plus ion concentration? Number one, the lumen has more H plus concentration because of the splitting of water molecules. So when water molecules split, pH2 is here. So splitting of water molecules is associated with pH2, right? So splitting of water molecule gives you more H plus ions inside the lumen of the thylakoid. Number two, reduction of NADP to NADPH plus H plus. This also adds to this. So we are having more concentration of H plus inside the lumen than in the stroma. Done? Okay. Third reason, there is electron transport chain, right? Inside the electron transport chain also we need some electrons. For example, it can be an exhibition for students, but we can also have student volunteers to help the teachers, right? Though it is electron carrier, you need certain electrons for it to work. So the electrons for the electron transport chain comes from the stroma. So there is some amount of electrons here. The stroma is willing to give it to the electron transport chain. So the electron from the stroma goes to the lumen along with the H+ carrier. H+ acts as a vehicle which carries the electron to the ETS. So when it carries the electron, again H+ accumulates inside the stroma, inside the lumen. Yes or no? Right? So H+ carries your electron. So in a way, H+ concentration is high inside the lumen than in the thylakoid. So when a difference in concentration than in the stroma. So when this gradient is set up, you have another player who's coming here. So can you see F0 and F1? So you have a complex called ATP synthase complex, which can actually synthesize ATP. So this has two components, F0 and F1. F0 is here, F1 is here. So F1 is peripheral, if you can see, right? So when the difference is there, now when the lumen and the stroma understand, yes, the lumen has high of H+ and I have low of H+, the gradient has to be broken, right? So the H+ ions pass through the F0. When they pass through the F0, there's a conformational change in the F1, so the H+ ions come out. When it comes out, the ADP is converted into ATP. So for every 2 H+, you have 1 ATP being produced. So the gradient has to be broken. So the H+ ions pass through F0. So when they pass through the F0, the conformational change of F1 is there. So the conformational change converts ADP to ATP. This is your chemiosmotic hypothesis. Is it easy? Yes? Did you guys understand what is chemiosmotic hypothesis? Any doubt? Clear? So thus, for the production of ATP, you should have protons, right? You should have a proton pump, because of which you have more H+ here. Then you should have a proton gradient. Then you should have an ATP synthase to break it, and first of all you should have a membrane between which there is a gradient. Alright? Fine. So a chemiosmotic hypothesis requires a membrane across which the gradient is set up, a proton pump so that there is more H+ inside the lumen. There should be a gradient, higher concentration and lower concentration, and ATP synthase. Okay, so this is what is important. This is what allows the formation of ATP. Similarly, NADPH is also produced. That is what scientists quoted. It is still a hypothesis because it is neither rejected or neither it is directly proved inside the cell. Right? So now what happens? Your stroma has ATP now. Right? So your ATP is produced in the stroma. So ATP is a product of light reaction. Now, as a result of light reaction, ATP is produced. Now your stroma is responsible for the dark reaction where the sugars are produced. Now, how this ATP is used in the production of sugar is the next question, which we'll address on Wednesday's class. Alright, how this ATP or NADPH produced is used in the synthesis of sugar is the next topic. Alright, so that is where your C4 and your C3 cycle comes into picture. So I will teach that on Wednesday's class. Alright, so I hope you understood the most important concept of chemiosmotic hypothesis along with what is cyclic and non-cyclic photophosphorylation. Alright, so I hope it is easy. So please solve some questions. So if you find any question tricky, so please type it here, or I hope you are connected with me in Telegram so you can take a picture and put it there, and I am really grateful to help you. Alright, so thank you so much for joining my lecture today. So hope you enjoyed. So I will meet you on Wednesday at 4:30. So until then, this is me signing off. So take care and bye.